So we have an incredible new treasure trove of information arising from the Cancer Genome Atlas and similar programs in the academic community. They have focused so far on the DNA and RNA level. We and a number of other groups have begun to provide a protein-based analysis of the cancer genome. This is providing new information and opportunities that have not been present previously. We know that the correlation between DNA, RNA, and protein in many cases is low, and importantly, protein function cannot be predicted well by looking at DNA and RNA. Yet we know that it is protein function that drives the behavior of the cell and is indeed the target of most of the therapeutic drugs that we use today. So combining information from the functional proteome, how the protein network is functioning, with what is happening at the DNA and RNA level will provide us with information that we've never had before. The challenge, and the big challenge that we have had, is that we've had approaches that will let us look at one or a few proteins at a time. But it's clear that we need to look at networks, we need to look at interactions between different protein pathways, and use that information. A number of different technologies are emerging that are beginning to allow us to look much more broadly at what is happening in the proteome. We've heard at this meeting or had presentations at this meeting on mass spectroscopy approaches. There are companies that are moving a technology that we use extensively called reverse phase protein arrays to the clinic. And we've developed, along with a collaborative effort with NanoString, a way to measure DNA, RNA, and protein on a single platform in a coordinate manner. So we will have the ability very rapidly to be able to add protein analysis to the comprehensive analysis of patient tumors that we're doing to allow us to predict outcomes better, to select appropriate therapies, and importantly, in an area that we've put a major emphasis on, to identify rational combination therapies that are likely to get around drug resistance.